Hello, Fall 2018 Bio 111. I am a tardigrade, also known as a water bear. I'm excited to be here at LMU to teach you a little about myself. My origin dates back to the Cambrian era, which was about 500 million years ago, when major groups of organisms began to emerge. The details of my origin are unknown, but because 17.5% of my genome comes from foreign species, scientists believe that I have gone under horizontal gene transfer in some way that led to my eventual creation. These are some of my defining characteristics. My scientific name is Hypsibius jardini. Most of us live in moist terrestrial or freshwater environments. I have plump body with 4 pairs of legs and claws. I range from 0.33 to 0.5 millimeters in length. I am essentially indestructible. I can live in harsh environments such as space, mountaintops, deep sea, and even the Antarctic. But I am mostly found in normal environments. I can live on bryophytes, lichens, algae, and other plants. But how do I withstand all kinds of environments? Cryptobiosis. This means I can reduce my metabolic activity almost completely to withstand harsh environments. I can stay in this state for more than 30 years. When living conditions improve, I can return back to my normal active state. Here's a little more about me. I have 2 eye spots and sensory whiskers, a body covered by a thin cuticle of chitin and protein, and no respiratory system. Some more features include an intestine for digestion, a mass of nerve cells in my head that makes up a majority of my nervous system. Let's talk a little bit about the diversity in the taxon I am associated with. There are 3 major groups within my taxon: Eutardigrade, Heterotardigrade, and Mesotardigrade. Eutardigrade are characterized by living in fresh water, the absence of lateral appendices, the ability to live in very dry environments through cryptobiosis. Heterotardigrade are characterized by presence of cephalic appendages as well as legs, slightly different anatomy of the reproductive system. Finally, mesotardigrade are characterized by only having one species, having 6 claws of equal length on each foot, and possibly having become extinct due to a natural disaster. Our reproductive status varies by species. Those that live on bryophytes reproduce asexually or through hermaphrodites. Those that live in marine environments reproduce sexually through 2 sexes. Otherwise, we reproduce all year round. When it comes to laying eggs, my legs are either laid freely exposed on a substrate or deposited on my outer skin after molting. To eat, we have stylets in our mouth that can pierce our food source. Our pharynx then helps suck the contents through it into our gut. We eat plant cells, algae, and bacteria, and each other. We move by using our legs in a stepping motion. We are protozoans, so our first developmental opening is our mouth. Our embryos develop through holoblastic cleavage, and we develop fully through molting or shedding in 5 to 10 days. We molt 4 to 12 times in our life, which can range anywhere from 3 to 30 months. More about molting. I molt more rapidly in my first few months because I'm growing so quickly. When I molt, I shed my old cuticle, including my claws and the lining of my foregut and hindgut. Unlike humans, I grow by enlargement of my cells, not multiplication. These are the stages of life I can go through. 1. Active. 2. Cryobiosis. 3. Osmobiosis. 4. Encystment. 5. Anoxybiosis. And 6. Anhydrobiosis. In any stage except active, I don't age or grow, making my lifespan longer. I get moved around to such different environments through wind dispersal with the mosses. Also, if I'm scaring you, don't worry. I can't harm you. Fun fact, chances are you might have even eaten me by accident before. We tend to end up on food that water is touched with, which is a lot of what you eat on a daily basis. And no, we can't even harm you from the inside. We don't survive during the digestion process due to the acid your stomach produces. Instead of harming you, science has shown that we can protect you from harmful radiation given off of X-rays. This is my placement on a tree looking at shared genes. My sister taxa include Echinodermata, Chordata, Annelida, Arthropoda, and Mollusca. I share a most recent common ancestor with the aforementioned. This is my placement on a tree looking at shared morphology. My sister taxa include Annelida, Chordata, Mollusca, and Arthropoda. I share a most recent common ancestor with all of them. This is my placement on a tree from literature. My sister taxa include Arthropoda and Onychophora. I am further removed from Mollusca than in previous trees. You may have also noticed this tree looks different than the morphologies tree. Good observation. This tree differs because this tree is organized by 18 srRNA sequences. The most likely reason for their difference is that some morphological similarities are not related genetically but are homoplastic. For example, I am related closer to Annalita on the tree created in lab because we share traits such as cephalization, symmetry, and digestion. But our genetic makeup still differs greatly. My placement on phylogenetic trees can be tricky because it is difficult to collect and culture me, but scientists are slowly figuring it out. Thanks for listening. I want to thank Ash, Dr. Hertz, Dr. Willett, Tanya, and the rest of the biology department for having me today.